Hello to all students. I hope you are doing fine. This is Professor Masood Fuzail. In this video lecture, we are going to discuss the protozoans. Protozoans are animal-like protists which belong to kingdom Prototista, which is a diverse kingdom of all kingdoms. It has so many organisms and so much diversity. But we are going to discuss only protozoa. Let's look at the meaning of the protozoa. If we look at the meaning of protozoa, proto means first formed, zoa means animal. So these are the animal, I think, which have been originated first of all in the whole kingdoms. So that is why they are known as protozoa. So we are going to discuss their general characters, their structure, their anatomy, their physiology, and their reproduction. So first of all, protozoans, which are also known as chemo organotrophic protists. Chemo organo means they can synthesize their own food with the help of chemicals like euglena, and organo mean they can obtain their food from detritus, organic detritus, which is decaying matter of animals and plants in the water, and they obtain their food from those molecules. That is why they are known as chemo organotrophic mean food. Protists are the animals which are known as protozoa. So these animals, the protozoa, also known as chemo organotrophic protists. Now the diagram already I have made just like before, and in the internal structure is showing in this diagram, and we are going to discuss characteristics of the protozoan one by one with the help of diagram. So let's look at the structure of the this animal like protozoan. It is composed of a single cell. Usually they are composed of single cell, but sometimes they form colonies which are very complex, just like coinoflagellates. So this whole animal, this whole diagram can cover all the animals present in this group, like amoeba, paramecium, which contain cilia, zooflagellates, coinoflagellates, and all the animals can be considered in this diagram. The outermost membrane of these animals is known as plasma membrane. These animals do not contain any cell wall made up of cellulose, peptidoglycan, or collagen. So these organisms are devoid of cell wall. So the outermost boundary of the organism is known as plasma membrane. Just beneath the plasma membrane, there is another layer which is known as pellicle. Pellicle is made up of microtubules. Microtubules are specialized tubule-like structure which are made up of tubulin protein, which forms a layer just under the plasma membrane. This pellicle gives the shape to the animal, just like a paramecium has a shape, amoeba has a shape, euglena has a shape. But this pellicle is also flexible. It's not very rigid structure. Inside the pellicle there is a liquid, as you can see in this diagram in blue color. The dots you can see in the diagram, this is a liquid medium which is known as cytoplasm. The cytoplasm of protozoan can be divided into two parts. The outer, which is peripheral part, which is non-granular, is usually known as ectoplasm, while the central cytoplasm, which is granular in shape, is known as endoplasm. So ectoplasm and endoplasm are also involved in locomotion in amoeba, and they also contain storage bodies and food molecules and waste molecules which are dissolved inside the cytoplasm. These animals usually live in fresh water as well as in marine water. The protozoan which are living in marine water, they usually have isotonic body. So their internal environment is isotonic to the external environment, so they do not have to regulate water. But the protozoan which are living in freshwater bodies, they usually regulate water, which is known as osmoregulation, because in freshwater most of the water tends to enter inside their body, and their body swells like a balloon, and if they do not regulate or osmoregulate the water inside their body, they will burst like a balloon and their existence will be very difficult. So that is why they keep an organelle which is known as contractile vacuole. Contractile vacuole is a specialized vacuole which is responsible for the osmoregulation in fresh water. So this vacuole moves inside the cell and absorb excess water with the help of the channels which comes out from the contractile vacuole. These channels absorb excess water, and when excess water has been absorbed in the vacuole, this vacuole attaches with the plasma membrane and throws out the excess water. So this vacuole constantly absorbs water from the cell and throws out from the cell with using the energy which is known as in the form of ATP. So this is very important organelle which is found in these animals or animal like protozoans. If we look at the digestion or the food, they usually live in a competitive environment where food is very restricted, and they usually eat dead organic matter of animals and plants body. So for most of the time they use their general surface for the absorption of the food in the form of diffusion, but sometimes they do endocytosis. With the help of endocytosis, the food enters through plasma membrane, where a specialized region from where food enters is known as cytopharynx, where food can enter inside the cell with help of endocytosis. So usually their food, if it's in the large particle, then it will enter as endocytosis, and if it is in the small particle, it can directly diffuses from water into the cell. When food enters into the cell, it forms a vacuole which is known as food vacuole. Food vacuole is surrounded by a membrane, and this food is being digested with help of enzyme present in the organelle known as lysosome. Lysosomes are attached with the food vacuole, enzymes are released into the food vacuole, and food is being digested. After digestion, food is distributed to all over the body, from where it is absorbed and assimilated. The rest of the food particles which are undigested, which are unnecessary, becomes egestion vacuole. This egestion vacuole, when needs to ingest the food from the body, attaches with the plasma membrane and form a structure which is known as cytopg. And from cytopg, excess or waste material can be thrown out from the plasma membrane. So cytopharynx for the entry of the food vacuole for digestion of the food vacuole, egestion vacuole for the elimination of the waste material, and cytopg is the structure from where food vacuole will throw out the undigested food material from the body. Usually the animal has a plasma membrane all over the body, which is usually act as respiration for gaseous exchange. Oxygen and CO2 are absorbed with the help of diffusion, and also ammonia, which is a nitrogenous waste material produced in the body, can also diffuse from inside the body to the outside environment. If we look at the kingdom Prototista, the animals which belong to kingdom Protista, they are very diverse. They are animal-like, they are plant-like, they are fungus-like. These animals which are studied under this branch is known as protistology, and the scientists who study specially protozoans, which are known as protozoologists. So if I am the zoologist and I am studying on these kind of animals, then I will be considered as protozoologist. Next point is Chemo Autotroph. Some of the protozoans, just like Euglena, they are photoautotroph. They can synthesize their food with the help of photosynthesis, with special type of photosynthesis which is known as Chemo Autotrophs. If we look at these organisms, these organisms live free life. They are free living, just like amoeba, and some live as a parasite, just like entamoeba, histolytica, and trypanosoma, which is act as a parasite, and they are pathogens and cause different types of diseases, like entamoeba histolytica cause dysentery, and trypanosoma causes the African sleeping sickness. Some animals, they usually protozoan animal live a symbiotic life. As you know that symbiosis is a relationship in which two organisms live together. There are different types of symbiosis. For example, parasitism, in which a host and parasite live together, just like a trichonympha, which resides in the gut of a termite, where it digests the wood for the termite, which is a symbiotic relationship between which one protozoan getting benefit while the other is not getting harm or getting benefit. While some other are very harmful to the body, they are producing different kind of diseases, and if they are causing diseases, that relationship is known as parasitism. If both of the partners are getting benefit, then it is known as mutualism. And if one get benefit and other do not get harm or do not get any benefit, that is known as commensalism. So they live as a parasites, they live as a mutualists, they live as a commensalists. So they have pretty different types of relationship in their environment. If we look at the reproduction, they can reproduce sexually as well as asexually. Usually they reproduce by asexual means of reproduction, but sometimes they also use sexual reproduction. If we look at the means of reproduction, the first important, which is most commonly used reproduction method, is binary fission. During binary fission, a single organism, just like amoeba, divides into two cells. In the first stage, its nucleus divides into two. After that, cytoplasm divides into two halves, and then animals divides into two animals. This is known as binary fission. Sometimes they also reproduce by budding. A bud is produced on the general body surface. Nucleus divides, and one nucleus moves into the bud with little bit cytoplasm. Then this bud is separated from the parent body and develop into new organism. Sometimes they also, under unfavorable condition like amoeba, they do schizogeny, which is known as multiple fission. A single nucleus divides into many nuclei along with little amount of cytoplasm, and they develop into spore-like structure inside a single cell. When favorable condition arrives, this whole cell bursts and many cells are dispersed inside the environment. So this is known as schizogeny or multiple fission. During sexual reproduction, they usually produce gametes in the form of cells like egg and sperm, and they fuse to form zygote and then develop into new organism. They also do conjugation, just like in paramecium, during which nuclei of two different strains of paramecium are exchanged to develop a recombination in these organisms. So that's all for today. I hope it makes sense, and I'll see you in the next lecture. Until then, bye.